Researchers at the University of Toronto have made a groundbreaking advancement in gene manipulation. By harnessing a bacterial immune defense system known as CRISPR, they have developed a technology that can efficiently and precisely control RNA splicing. This innovation opens up new possibilities for understanding gene functions and correcting splicing deficiencies that contribute to numerous diseases and disorders. CRISPR, or clustered regularly interspaced short palindromic repeats, is a bacterial immune defense system that has been adapted for gene editing. In this study, researchers used a catalytically deactivated version of an RNA-targeting CRISPR protein, known as dCASRX, to control RNA splicing. RNA splicing is a process where coding segments called exons are joined together, and non-coding segments called introns are removed. Almost all human genes produce RNA transcripts that undergo splicing. Exons can be alternatively spliced, which diversifies the regulation and function of approximately 20,000 human genes that encode proteins. This allows for the development and specialization of different cell types. However, the functions of most exons and introns remain unclear. Misregulation of alternative splicing patterns is a frequent cause or contributing factor to various diseases, including cancers and brain disorders. Existing methods for precise and efficient manipulation of RNA splicing have been lacking. Researchers have struggled to find tools that can accurately target and modify specific exons without causing off-target effects. This has limited the ability to study gene functions and develop therapies for diseases caused by splicing defects. The new technology developed by the University of Toronto researchers addresses these challenges. In their study, the researchers joined dCASRX to more than 300 splicing factors to discover a fusion protein called dCASRX-RBM25. This protein can activate or repress alternative exons in a targeted manner. Our new effector protein activated alternative splicing of around 90% of tested target exons, said Daiyang Li, the first author of the study. This multi-level manipulation allows for the experimental testing of functional interactions between alternatively spliced variants from genes, determining their combined roles in critical developmental and disease processes. The new tool has a broad range of applications, from studying gene function and regulation to potentially correcting splicing defects in human disorders and diseases. Our new tool makes possible a broad range of applications, from studying gene function and regulation to potentially correcting splicing defects in human disorders and diseases, said Blencowe, principal investigator on the study. This technology could be used to treat numerous human disorders and diseases, such as autism and cancers, where splicing is often disrupted. We have developed a versatile engineered splicing factor that outperforms other available tools in the targeted control of alternative exons, said Mikko Taipale, another principal investigator on the study. It is also important to note that target exons are perturbed with remarkably high specificity by this splicing factor, which alleviates concerns about possible off-target effects. The researchers now have a tool to systematically screen alternative exons to determine their roles in cell survival, cell type specification, and gene expression. This technology has the potential to revolutionize the way we study and treat diseases caused by splicing defects. Supported by the Canadian Institutes of Health Research and the Simons Foundation, this research paves the way for future clinical applications that could significantly improve patient outcomes in various genetic disorders and diseases.